Today topic of discussion is mycoplasma. What is mycoplasma? Myco means fungus like filaments. Plasma means plasticity of safe. So now we will discuss about that morphology of mycoplasma. Mycoplasma is a group of bacteria which are devoid of cell wall. That means cell wall are highly pleomorphic in nature. They are called stable L form. L stand for Lister Institute, London. Mycoplasma are called Pneumonia like organism, PPLO. Here the cells are bounded by soft trilaminar unit membrane which contain sterols which give the plasticity. Mycoplasma is a genus in which species are gram negative and they are stained better by Giemsa stain. The stain used for mycoplasma is Giemsa stain. The members are non-sporing, non-flagellated, non-motile, but some species have gliding motility and also do not have fimbria. This is the morphology of mycoplasma. Next, coming to antigenic properties. It is a pneumonia like organism, gram negative, special staining we are using here, Giemsa stain. Then surface antigen include glycolipid and proteins. Glycolipid are identified by complement fixation. Then protein antigen are identified by ELISA. So these are the main antigenic properties in mycoplasma. Surface antigen having glycolipid and protein. Glycolipid identified by complement fixation. Protein by ELISA, enzyme linked immunosorbent assay. Next, pathogenicity. It causes two main type of diseases. First one is pneumonia, second one is genital infection. First see about that pneumonia caused by mycoplasmal pneumoniae. The mycoplasmal pneumonia, which is called primary atypical pneumonia. This typically presented as tracheobronchitis in 1/3 of the patient. It can lead to pneumonia. First one is mycoplasmal pneumonia, also called as primary atypical pneumonia, presented in tracheobronchitis. 1/3 of the patient can lead to pneumonia. Then clinical features of pneumonia. It has gradual onset of fever, malaise, headache, sore throat, paroxysmal cough with blood tinged sputum. These are the clinical features: fever, malaise, headache, sore throat, paroxysmal cough with blood. Then radiologically can be visualized as patchy consolidation involved in one of the lower lobes. Radiologically we can visualize patchy consolidation. Then diseases. Diseases caused by mycoplasma usually self-limited. There can be some complication, rarely, which otitis media. It means infection of middle ear, then bullous myringitis, which is severely painful, and a small fluid filled blister formed by eardrum. It causes rashes, meningitis, encephalitis, hemolytic anemia. These are the complications of Mycoplasma pneumoniae. Next is second infection or genital infection. These are caused by Mycoplasma hominis. This one is caused by Mycoplasma hominis. It is transmitted by sexual contact. It can lead to, in males, urethritis, bronchitis, then Rider syndrome. This Rider syndrome, which is reactive arthritis in case of male. This Rider syndrome is a triad of it causes conjunctivitis, urethritis, and arthritis. Genital infection in male, it causes mainly urethritis, bronchitis, and Rider syndrome. In the case of female, the genital infection include acute salpingitis, that means inflammation of fallopian tube, then pelvic inflamed disease, then cervicitis, that means inflammation of cervix, then vaginitis, inflammation of vagina. It also associated with infertility, abortion, postpartum fever, and low birth weight of infant. This is pathogenicity of Mycoplasma. In female, it causes acute salpingitis, pelvic inflamed disease, then cervicitis, vaginitis. Then also associated with infertility, abortion, postpartum fever, and low birth weight of infant. Next one is laboratory diagnosis. Now coming to lab diagnosis. First one is collection of specimen for isolation. Specimen collected are throat swab and respiratory secretion. Second is isolation. Culture is done on complex media like PPLO broth, Pneumonia like organism. Here we are using 5 basic medium, it containing solid and liquid, in which it shows that fried egg appearance of Mycoplasma. That colony look like fried egg. Next, growth screening can be done by tetrazoleum reduction test, in which the colorless tetrazoleum is reduced by Mycoplasma into red color formation. So it will change to red color formation. Next one is molecular method, PCR assay. PCR is the rapid and specific test. Next method is the serology. It consists of 2 sets of tests. First one is specific, second one is non-specific. Specific test, it included immunofluorescent, then heme agglutination, then growth inhibition test. Non-specific, it included cold agglutination test, indirect comb test. Finally, treatment of choice, tetracycline and doxycycline. These are all about Mycoplasma. This one is the notes format. Introduction part, Morphology. It is a gram-negative one. Giemsa staining we are using. Then Androgenic property. Surface antigen having glycolipid and protein. Pathogenicity, it causes pneumonia and genital infection. First one is pneumonia clinical feature, then diseases self-limiting only. Complication, then genital infection caused by Mycoplasma hominis. Then female genital infection, laboratory diagnosis. Finally, treatment. Thanks for watching.